In this video, we'll talk about the very initial stages of development. We're talking about prenatal development, which is occurring before birth in utero, and neonatal development, which is occurring in newborn infants. Like most topics in this course, we won't be going into too much depth, but you'll just get a taste of the topics related to early development. When we talk about prenatal development, we're talking about the period from conception to birth. We're going from one cell to 200 billion cells in about 266 days. That's an average. This process of rapid development, in fact, it's our most rapid period of development as humans, is guided and shaped by genetic and environmental factors. We'll leave the genetic factors for a developmental biology class, but here we're going to focus on the environmental factors. The environment in utero plays an impactful role on the developing embryo. One way we observe this is through the effect of teratogens. A teratogen is a biological or environmental agent that negatively impacts development. This word teratogen stems from the Latin terra, meaning monster. And these can be substances like drugs or pollutants or toxic chemicals, or they can be conditions like severe malnutrition, extreme stress, radiation. And these can have significant negative effects on the embryo. For example, you might have heard of the negative effects of tobacco or alcohol exposure on a developing fetus. With tobacco exposure, for example, we see significant effects on the environment that the fetus is developing in. For example, we see reduced blood flow to the fetus, we see reduced transfer of nutrients, and we also see reduced oxygen levels in the mother's blood, which will correlate with reduced oxygen provided to the fetus. And so possible effects of tobacco exposure in utero include miscarriage, premature birth, low birth weight, low IQ, cancer, higher SIDS risk. SIDS is sudden infant death syndrome, which is a disease that affects newborn infants, leading to sudden death for unexplained reasons, and slowed growth. With exposure to alcohol, particularly at high levels, we see effects on the developing fetus. One main effect is we see disruptions in brain development. And this is partly because alcohol can actually cross the placenta and enter the fetus blood supply. We also see hyperactivity in children, intellectual and attentional difficulties, and facial deformities as a result of alcohol exposure. These are all symptoms of what we call fetal alcohol syndrome, or FAS, and this occurs after heavy drinking by a pregnant woman. The impact of a teratogen on a developing human is affected by several factors. One is the timing of exposure to that teratogen. So some developing structures in the body are more vulnerable to damage at certain times. There are these critical windows where exposure to a substance will lead to maximum negative effect. Two is the amount of teratogen that the fetus is exposed to. So low levels of a teratogen will usually have limited effects, and as we see levels increase, we're going to see more significant effects. Looking at alcohol consumption during pregnancy, one study by Kelly et al. That came out in 2010 looked at the outcomes associated with pregnant women who had consumed 1 to 2 alcoholic drinks per week during pregnancy, and they found that there was no noticeable effects on their children that they followed up with at 5 years of age. Third, the genetics of the individual can affect the impact of the teratogen. We see evidence for this when we look at fraternal twins who are exposed to the same teratogens, but we see different outcomes. And finally, we see that the sex of the individual can affect the impact of the teratogen. And we see that males tend to be more susceptible than females to damage by teratogens. And this is perhaps due to the male's Y chromosome, which is shorter and contains fewer genes than the female's X chromosome. So a genetic mutation caused by a teratogen in a male might have a relatively larger effect due to that shorter chromosome with fewer genes. So all of this about teratogens gives us another example of the interacting role between genetic and environmental factors, that whole nature versus nurture discussion. This is a classic example of epigenetics, that is, environmental factors that can affect gene expression. So coming up in class, we'll continue our look into development, looking at neonatal development, what happens right after birth. We'll also look at stages of development throughout childhood and into adulthood. And we'll look at language development as an example of a cognitive process that develops over our lifespan.